Exam 4 Study Guide. 4 events of cell division. Firstly, the reproductive signal is signaled, and it initiates cell division. It can be intracellular or extracellular. Next is the replication of DNA. Third is segregation, the separation of DNA into the two cells. And then fourth is cytokinesis, which is the separation of cell materials into the two cells. Prokaryotic cell division is called binary fission. It takes place in the 4 steps listed above, those being the reproductive signal, replication of DNA, segregation, and cytokinesis. A prokaryotic cell will divide when triggered by an extracellular signal, such as environmental conditions. Then it will replicate its DNA. This occurs through a replication complex, beginning at the ori and ending at the ter, which are the origin and terminus, basically, the beginning and the end, to remember it easily. After this, the segregation occurs by the ori regions moving to opposite ends of the cell, and then in cytokinesis, the membrane is pinched in, and the protein fibers form a ring. The difference between eukaryotic cell division and prokaryotic in terms of when it occurs is that prokaryotic cells will constantly divide so long as conditions are correct, while eukaryotic cells are part of a greater organism and only divide when a signal from elsewhere in the organism tells them to. Sister chromatids are recently replicated chromosomes that are still closely associated with each other. Mitosis is the process that separates 2 recently replicated chromosomes into 2 new nuclei. The cell cycle has 3 broad phases. These are interphase, mitosis, and cytokinesis. Interphase has 3 subphases. These are G1, S, and G2. The first of these is G1 phase, which is the first where the cell has not yet begun the process. Once it enters S phase, the DNA replicates, and the sister chromatids stay together. And in G2, the cell prepares for mitosis. To enter S phase, however, the cell needs to pass the restriction point. The restriction point is what prevents cells from constantly dividing. The factors involved in passing the restriction point are cyclin, CDK, and RB. Cyclin is an allosteric regulator, a type of enzyme, which binds to CDK when it needs to be activated. CDK is cyclin-dependent kinase, which once is activated can be used to phosphorylate RB. RB is the retinoblastoma protein. Normally, it is active and inhibits the cell cycle. However, it is deactivated by CDK phosphorylating it, allowing the cell cycle to occur. G0 phase cells are cells that are not in the cell cycle. To be activated, they must be stimulated by growth factors. These growth factors will activate pathways that end with cyclin production. There is a lot of DNA, so it needs to be packed together by proteins called histones. Nucleosomes are bead-like structures that are formed as a result of this interaction between DNA and histones. A chromatin is a DNA module bound by proteins. A chromosome is a long, thread-like structure made up of DNA and proteins that contains genetic information. If the chromosome is in a X shape, that means it is the metaphase of mitosis. Cohesins are proteins that hold sister chromatins together during G2. The centrosome is the region of the chromosome that acts as a site for sister chromosomes to connect. Also, it is the middle of the chromosome, so it's the little, most pictures, the dot in the middle that they all connect to. The first phase of mitosis is prophase. In this phase, the chromosomes condense, the spindles form, and the centrosomes move to opposite sides of the cell, and in late prophase, kinetochores form, which are vital for movement of the chromosomes. Then there is prometaphase, in which the nuclear envelope is broken down. All cohesins are removed, except for the centromere, and the chromosomes attach to the spindles. Then is metaphase. Then the chromosomes align themselves along the middle of the cell, called the equatorial plate. Then anaphase occurs. This causes the separation of the chromosomes. They are referred to as daughter chromosomes after the separation. These daughter chromosomes begin migrating to the poles, opposite ends of the cell, and following this is telophase, in which the nuclear envelopes and nuclei reform and the chromatin decondense. APC is important as it hydrolyzes cohesin, allowing the chromosomes to separate. In animal cytokinesis, proteins cause the pinching of the cell membrane that splits the cell in two, while in plant cytokinesis, vesicles appear along the plane of cell division, creating new plasma membrane. A gamete is a sex cell created by meiosis. Somatic cells are body cells not specialized for reproduction. Homologous pairs are matching pairs of chromosomes that carry the same genes for the same traits. Haploid means half the chromosomes, 23, while diploid is twice as much, 46. Fertilization is the combination of two haploid gametes to form a diploid zygote. A zygote is created by two gametes, also known as a fertilized egg. Zygotes grow through mitosis, not meiosis. Cells that undergo meiosis are specialized cells called germ cells. In males, they produce sperm cells. In females, they produce egg cells. Meiosis occurs in two broad steps, meiosis 1 and meiosis 2. The first step of meiosis 1 is prophase 1, in which the chromatin begins to condense. The next part of prophase 1 is prometaphase, where crossing over occurs. This is where non-sister chromosomes exchange genetic material, giving more varied genetic results. Next, in metaphase 1, homologous pairs line up on the equatorial plate. This is different than mitosis, as it is the pairs line up instead of just the chromosomes. Then, in anaphase 1, the homologous chromosomes, each with two chromatids, move to opposite sides of the cell. And in telophase, the chromosomes gather into nuclei, and the original cell divides. Then meiosis 2 begins with prophase 2. In prophase 2, the chromosomes in the two sister cells condense but do not replicate. Then in metaphase 2, the centromeres, remember the middle of the chromatids, line up along the equatorial plate. These are then pulled apart into chromosomes in anaphase 2. Each new cell has a unique genetic makeup due to crossing over and random assortment. And finally, in telophase 2, the chromosomes gather into nuclei and the cells divide, resulting in 4 daughter cells. And each daughter cell has a haploid number of chromosomes. Non-disjunction is an error that can occur during meiosis. This occurs when homologous pairs during anaphase 1 or sister chromatins in meiosis 2 do not separate correctly. This can result in too few or too many chromosomes. In humans, having an extra chromosome 21 causes Down syndrome. Mitosis and meiosis are both processes of cell division, but they differ significantly in purpose and outcome. Mitosis is used for growth, repair, and asexual reproduction, producing two genetically identical diploid daughter cells. It involves one round of division and does not include crossing over. In context, meiosis is used for sexual reproduction, producing 4 genetically diverse haploid gametes, sperm or eggs, through 2 rounds of division. Meiosis includes crossing over during prophase 1, which leads to genetic recombination. Mitosis produces somatic cells, while meiosis produces gametes, and the latter is essential for maintaining genetic diversity in offspring. Apoptosis is programmed cell death. It is important as old and damaged cells can cause damage to the body. Signals to initiate can include hormones, growth factors, viral infections, toxins, extensive DNA damage. A tumor is a large mass of cells. Benign tumors grow slowly, resemble the surrounding tissue, and remain localized, while malignant tumors do not resemble the parent tissue, and assumably also do not remain localized. A gene is DNA that encodes info to build proteins. Gregor Mendel is the father of genetics. The phenotype refers to the physical appearance of an organism, while the genotype, the genotype is the genetic makeup of the organism. Dominant alleles always express their proteins, while recessive don't whenever a dominant is present. Homozygous refers to having two of the same alleles, either dominant or recessive, while heterozygous refers to having one of each. There is no such thing as heterozygous dominant or heterozygous recessive, they just have one of each. P refers to the parent generation, or the first generation of plants Mendel worked with. F1 then refers to the first generation of offspring, and F2 to the next generation of offspring after that. Offspring inherit alleles from both the mother and the father. Gregor Mendel had two laws. There is the law of segregation. The two copies of the gene separate during gamete formation, and each gamete receives only one copy. And the law of independent assortment. Copies of different genes assort independently. Punnett squares are easy. Remember how to do them. Line up the set of genes on top, and line up one set of genes on top, one on the bottom. 3-to-1 ratio for two heterozygous, the 1-to-1 ratio for a heterozygous and a homozygous recessive, 9-3-3-1 for two double heterozygous. You got this, it's easy. Remember the two most simple statistic rules, the multiplication and addition rules. This is brain dead easy. If you fail this on the test, you're dumb. Don't screw it up. It's very easy. The wild type is the allele present in most of the population. A mutation is a stable inherited change in genetic material. Incomplete dominance refers to when alleles are neither dominant or recessive, while codominance occurs when more than one allele encodes a functioning protein. A locus is a specific point on a chromosome. Pleiotropic refers to one allele with multiple phenotypic effects, and epistatus refers to the expression of one gene being influenced by another gene. Linked genes are genes that are close together on the same chromosome, which usually travel together during crossing over. Light, temperature, and nutrition can all affect the expression of the genotype. Siamese cats are a great example of this, as their extremities are darker in color because the enzyme that produces the dark color is more productive in low temperatures than high temperatures. And obviously the extremities are at a colder temperature than the rest of the body. And prokaryotic cells exchange cells through conjugation. Plasmids are circular chromosomes in prokaryotes. These plasmids contain genes for unusual metabolic processes, antibiotic functions, and for making a sex pilus, whatever that is. Alright, and there you go.